talking about DNA. And hopefully you all had a chance to look at the video about Watson, Crick, and Franklin, and you got to know what they all did in the process of discovering DNA and how important that discovery was. You hopefully remember also what DNA stands for. So let me write that up one more time. DNA is deoxyribonucleic acid. So from that word, the D and the N and the A is where we get the name DNA. Okay, so here's what was so amazing about the discovery of the DNA, other than the fact that it was sugar. It was the fact that it had this structure that we call the double helix. So when you take the DNA off of the chromosome, remember we have these little chromosomes inside the cell's nucleus, and those chromosomes look something like that. When you unwrap those chromosomes, you've got these long strands of DNA that have a double helix. Sort of something like that. Let me blow that up bigger so we can look at that a little bit more closely. So the double helix looks something like this. When we say double helix, this is what we mean. So a double helix is another way of saying that something looks like a twisted ladder. So you can put that in your notes or however you're keeping track of information. Hopefully you've got a notebook going. So this is the twisted ladder is what that word double helix means. So if you could unwrap this ladder, what you would see is that you've got two different rails, and between each rail you've got these rungs. Okay, rungs like this. So if this is the railings of your ladder, if you're going to climb that ladder, and you've got these rungs in between. The railings of the ladder are made of that ribose sugar. Remember, we've talked about the fact that there's all sorts of carbohydrates out there, from the simplest glucose all the way up to more and more complex carbohydrate sugars. And this is a fairly complex sugar, but it's a sugar nonetheless. So it's got that same C6H12O6 basic structure of carbon, hydrogen, and oxygen. The ribose sugar has two strands, and that's where we get the D, or the 2 prefix for DNA. So there's two strands. There's strand 1 and there's strand 2 of the sugar, or the railings. Now the rungs. The rungs is where we get the nucleic acid word. So each of these rungs is made up of a different type of nucleic acid. In fact, each of the rungs is made up of a pair of nucleic acids. So these are a pair of nucleic acids. Those pairs of nucleic acids have a number of different names. They go by a number of different names. I'm going to put their names over here. So what I'm talking about are the nucleic acids. Redraw that arrow here. Talking about these here. Those go by the name nucleotides, or sometimes called nucleobases, or sometimes just bases, DNA bases. So you'll see these three different words used kind of interchangeably in science texts. You've got the nucleotides, the nucleobases, or just the bases the DNA bases. And so there's always two of them here in the middle. This one on this side connects to this one. If you remember in the video, in James Watson and Francis Crick noticed that their chemical structure allowed them to be held together in a certain way with hydrogen bonds. So what's holding them together in between there is a bond. There's a hydrogen bond right there holding this nucleotide to this nucleotide. The interesting thing about that bond is it's actually a fairly weak bond that holds it together because it needs to be able to unzip and zip back up again. We'll get to that when we talk about DNA replication and DNA transcription in one of the upcoming days. Anyway, these nucleotides and nucleobases, there's actually 4 different nucleic acids that make up the nucleotides or nucleobases. There's only 4. And that was actually pretty shocking to scientists when they discovered that there were only 4. And I'll tell you why. Okay, so the 4 nucleotides and nucleobases, I'm going to rewrite this here. The 4, we're going to use just the word bases now. But remember, that means nucleotides or nucleobases. The 4 bases are adenine, thymine, guanine, and cytosine. Okay, there's those 4 nucleic acids that are the bases of DNA. There's only those 4. And here's the interesting thing. Adenine always pairs up opposite of thymine. So you've always got like an AT pairing. And guanine always pairs up with cytosine. So, like on the next rung, you might have GC pairing. Now, not necessarily in that order, though. You could have like thymine pairing with adenine, and you could have cytosine pairing with guanine. But you can never have adenine pairing with cytosine or guanine pairing with thymine. That doesn't happen. You've always got the ATCG combination. So adenine pairs with thymine, or thymine-adenine, either order, and guanine-cytosine, or cytosine-guanine, like that. So you've always got those particular pairings. Let me flip this DNA on its side now for a minute. Okay, so here's the same double helix flipped on its side. Here's our ribose sugar rails, and here's the rungs, like this. And we've already said that these rungs are made up of the nucleotides, or nucleobases, or we'll just call them bases. Okay, so over here, I'm going to draw different symbols of how they connect together. So like this, and like that. Okay, so we're just going to look at the bottom part now. Adenine, we said, always pairs with, do you remember? Thymine. And cytosine always pairs with guanine. And that means that thymine pairs with adenine. And that means that guanine would pair with cytosine. Right, okay, so you've always got that. Now notice down here then that we've got, we're just going to read the bottom right now. Notice right now then we've got these letters in a row. Now actually I've written 4 letters up here, but the most important number is 3. 3 letters. Every 3 letters of DNA is called a codon. So this is called a codon of DNA. Codons are one of the most important units of DNA. if we compare DNA to a language, we might think of the bases as being like letters. Okay, so here we have this alphabet, and it's only got 4 letters in it. It doesn't have 20-some letters. It's got 4 letters. That's it. Now, that might seem really surprising, but remember, computer code really is based on zeros and ones. That's an alphabet with 2 letters. It's just how you combine them together that's important. So in this case, the bases, or the nucleotides, are the letters of DNA. Then that means that the codons of DNA are like words. So if the bases are letters, the codons are words. Okay, if you were in an English class, what would come next? You'd have the letters, and then the words, and then words go together to make complete ideas. And what would a complete idea be called in English? It'd be called a sentence, right? Okay, so in the language of DNA, if we've got codons or words, then the next thing is what we would call a gene. A gene is a complete idea. It's like a sentence. What is an idea for? Well, it's not an idea for as much as the plans for making one protein. So ultimately what's happening here with DNA is that these are instructions for making proteins. The DNA makes the proteins actually go do the work. The work might be forming bones, it might be forming red blood cells, it might be forming some other portion of your body, it might be processing something or engaged in some other activity that your body is doing. The DNA is making the proteins by combining bases into codons and codons into genes. So one gene makes one protein. And just to kind of wrap this idea up, and so if we've got bases or letters, codons are words, genes are sentences, we've just kind of come back full circle, because if we're reading a book, we might call like a whole bunch of sentences together, paragraphs, yeah, and we do have genes that kind of work together to make sets of complex proteins, so they're kind of like paragraphs. But the next unit in English would be a chapter. So a book is usually divided up into chapters, and that brings us back to this guy. The chromosomes. So, the chromosomes are like the chapters of a book. That means that humans have 23 chapters in our book of how to make a human. Those chromosomes then all together make up the instruction book for making one complete human being. And then that means that the entire set of DNA, the genome, which is all of the DNA together, so the human genome is all of the DNA that makes a human. That genome is like the book, the instruction book, to make one complete you. And that's all I've got for today. So stay tuned tomorrow as we look at then how this DNA splits open to